Is the audio clear? Once again, please.
Yes, sir, it is.
Okay, thank you. Welcome back to the lesson on human embryonic stem cells. We were discussing about the derivation of human embryonic stem cells. We discussed about properties, self-renewal, and we discussed about differentiation, how the potency or plasticity is related to, or the correlation between differentiation and plasticity. With increasing differentiation, there will be decrease in plasticity. And we also discussed this in this whole course. There are 3 points. The first is about how to get the embryos, and then second one is how to derive the embryonic stem cells from them, and the third is differentiation. We learned about IVF and other ways, and how these eggs are usually produced or formed. This we are talking in terms of human embryonic stem cells. If it is other other organisms, usually we don't care as much as we do. We might some people would kill a rat, a mouse in experiments as part of science. That's a normal thing that happens. So we were also discussing about how to derive the embryonic stem cells. We discussed about whole embryo method, immunosurgery, and mechanical separation of the ICM. Whole embryo method is where we take the whole embryo, use pronase to get rid of zona pellucida, and then put the naked blastocyst onto the feeder layer. Feeder layers are irradiated with mitomycin C or irradiated or treated with mitomycin C to prevent them from dividing any further. They serve purposes of producing several biochemicals that might be useful for the survival of the cells that might arise from the blastocyst. They also provide sites of attachment. So medium contains something like this, Dolveco minimal eagle medium, and we're supplemented with fetal calf serum, fibroblast growth factor, and so on. So in about 2 weeks or so, they will grow, and you can differentiate those that are the cells that are derived from trophoectoderm, which are usually like something representative like this with high or low nucleoplasm to cytoplasm ratio, whereas the ICM from here grow as a lump and with a high nucleoplasm to cytoplasm ratio, and then these are removed from this plate and subcultured, and that is how an embryonic stem cell line is established. Immunosurgery method is nice. Same treatment. We'll treat with pronase and then treat with anti-human antibodies if the embryo was of human antibody. The picture depicts of mouse, so they use mouse antibodies. When you say rabbit anti-mouse antibodies, that would mean that these antibodies were raised in rabbit against mouse antigens. Okay, they typically would take a rabbit and inject some of the mouse proteins or so. So the rabbit will generate antibodies against the mouse, and then they take the serum of the rabbit and isolate the antibodies and use it for further purposes, as in this case. So they treat the whole blastocyst, the naked one, after removing the zona pellucida with the antibodies. So the antibodies will coat, they will bind everywhere, and then we add guinea pig complement. The complement system would get activated upon binding to the constant regions of the antibody and will make pores on the trophoectoderm cells. ICM is inside, so it's a little bit protected. And after that, there are several washings, and upon washings, there could be a washing here also to remove excess antibodies. Here, there will be a washing to remove all the excess complement. And then over time, you'll have the trophoectodermal cells lysed, and then the ICM is taken and cultured or subcultured on feeder layers. And that is how the new cell line has been established, embryonic stem cell line. So the microdissection, the mechanical ways is to take the embryo, cut it into pieces, and then take out the ICM. We will, towards the end of this into this class, we will probably see the videos, or else in the next class. Okay, but the instrument is called as micromanipulator, and then people do in vitro fertilization or any manipulation of the embryos or evaluation of the embryos under the micromanipulator, which is basically a microscope with a lot of things that you can fine things to manipulate embryos, which are really tiny. So the next part is today we are going to discuss a little bit about differentiation. What we are going to discuss are general themes. It's not necessarily the everything. Say if you want to learn neuronal, how to differentiate embryonic stem cells into neuronal progenitor cells, you would have a protocol. If you want epidermal, there is a different protocol. If you keep on thinking, there is a whole list of protocols. So what we are discussing right now is about the general practices or general things that we need to know about differentiation. So the first thing is, say there are typically or traditionally speaking, there are two steps. One is from embryonic stem cells, they generate something called as embryoid body, which is kind of analogous to an embryo that is growing in the womb, or which is derived from ICM. So it is something like that. They make embryoid bodies. From embryoid bodies, they try to derive different cell types. It's not necessary that they always follow the same scheme, but this is traditionally the scheme that people have followed quite successfully. So what is an embryoid body? The first thing we'll say, it is a spear-like cells. It's spear-like structure with a lot of cells. And those cells are trying to live their differentiation fate, right? They have already differentiated from morula to as a zygote-like cell from the morula to ICM in the blastula. That means their fate, differentiation fate, has been already determined, and they have to undergo the formation of the primordial germ layers. So this is an embryoid body. Embryo would have been in the natural one. Embryoid means embryo-like body. So there are some ways to make embryoid bodies. That is by hanging drop method. We will see in the next slides, and where as we will see, in the drop the cells are inverted, right? I mean the drop is present, which will discourage the attachment of the cells to the cell plate. And then what will happen is they will try to bind to each other, and they will form clusters or masses like this. And these masses, when they are allowed to grow, they keep dividing, and then they will develop several the primordial germ layers and so on. Okay, like similar cell differentiation. So this is usually the first step in trying to differentiate different types of cell lineages from embryonic stem cells. Here is a method. We will see a few more methods, of course. And this is a Petri plate, a cross-section of it. And they have put several drops of media containing embryonic stem cells, the dotted ones. And then they would invert the plate. So the drops were on the bottom plate. This is the cap that is on the top. And when we invert, you will have the cap on the bottom, and the bottom is up. And now the cells will not, they at least because of gravity or so, you can assume that the cells will come to the bottom of this hanging drop. This drop is hanging, so you call it as hanging drop method. They also put a small trough or some liquid water, especially mostly. It is to maintain the humidity within these. If there is no humidity, the droplets could start evaporating. And if they evaporate slowly, the droplet will become smaller and smaller, which means it's also changing, so different concentrations of different compounds that are present and so on. It's not healthy. So to maintain that any loss of water, or to maintain the humidity, if there is a lot of humidity here, the droplets are unlikely to evaporate. So, and over a period of time, say 2 days or so, the cells that are present, they will clump together and form some structures, which are kind of early embryoid bodies. And once they are formed, you can invert them back and put them in culture, and they grow as if they are embryos, or trying to develop different lineages of cells with differentiated cells within them. And then each one can be put into microtiter plates, which will be there. We will see in the next slide. So that is one way of doing. The method is called as hanging drop method. There are this is how the plates would normally look like with these drops. That means we need to be very skillful, very steady hands, or actually deft hands, to keep them. Right? So there is also one more thing together. That's always a risk. And if it mixes up, or the whole plate might actually get spoiled as well. That is one of the technical risks that is present. There are other ways also where people use. They first they make a comb using some gels, and then you can make a comb. That comb can be used to make the micro wells in agar plates like this. And when you pour, I think if you are coming back and we do molecular biology lab, you will do electrophoresis, and in that time you will see how do you mold things. We are not going to do exactly this one, but if you know electrophoresis, or if you understood or studied, we'll be making agarose gels with wells where we will load the samples. During that time, to make wells, we use comb, something similar, only that the shape is different. For the purposes of embryoid body formation, they use a similar technology, and they use agarose, which may not be very good attachment site for the cells. So when you put the culture media along with the cells, they form embryoid bodies within wells, and they keep growing, and then we can grow them in suspension culture to grow as independent embryoid bodies. This is another way of how to generate embryoid bodies. The third, or the best one, is something like this, where you want to do a lot of things, and this is a kind of advanced technology in that. So there is a special designed microtiter plate like things, where you have several pores like this. So this is the cross section. If you understood this, it will be nicer. And you, as I discussed in the previous slide of in here, there is an inversion steps like this. And if you are inverting, there is always a problem of mixing. How to generate hanging drops without these steps of inversions? That's the question here. So for this, they designed a microtiter plate with the cross section. This is the cross section, and here you have tiny pores in the plate. Here is another pore. Here is another pore, like that. So if you see the close shot that is shown in C here, the researcher can just directly deposit the culture here, because there is enough contact area. The drop would already drop will hold on like this, and it will form a drop. It will form a hole because of its own surface tension. It can hang on to this surface here. Although there is a pore, it can still hang on. About 10 to 20 microliters can do that. If you overload it, then of course it will fall into the bottom. And then then slowly the cells will aggregate, and then they form into a spheroid, which can be used, or embryoid in the case of embryonic stem cells. Interestingly, many cancer cells also, if not all, if you put them, they can also form something like this, spheroids. And I think people use it in cancer biology as well. So the rest of the things are similar. Here you have the droplets, and you will allow the culture. And here you're putting water, so that distilled water, to prevent any evaporation from the water will maintain the humidity, so that the droplets will not get evaporated or shrunk because of water evaporation from the droplet.
So, what is the importance of the hanging? Because why couldn't we do it on a plate like normally how we culture bacteria and all?
Yes. So this is the first thing. Is the point you have to remember is gravity is there, and then they will settle and attach to the bottom of the plate. If it is in this case, say for example, if you leave it like this, right? That's the question. Why to do all this stuff, hanging drop? The cells will settle down because of gravity, and they will form. They will start interacting with the surface of the glass plate or whatever plate we are using. Nowadays we have plates that some of them can allow contact, some of them cannot. The point is, for an embryo in general, it is to be something like a sphere. So if you keep it as a hanging drop, that is an ideal position. It is ideal for formation of a sphere. Whereas you cannot say the same, why should cells grow as a sphere, or how will they grow here? It will rather be something like a flat lump. Okay. Whereas if it is in a hanging drop, the conditions are very good for complete formation of a sphere. That is the reason. Okay.
Okay, so we are basically providing something similar to the natural conditions they are
That's
multiplied.
right. Exactly. There are no pressures on any one side. Okay. Yeah, that's right. Okay, that is about hanging drop method. So the next ones will be about how to derive and derivation of propagating. We will leave this. This is just a picture of showing different cells, how would they look like, and so on. So this is embryonic human hESCs, or embryonic stem cell colonies, are growing on feeder layer. This would be the feeder layer, and these are colonies of human embryonic stem cells. This is a blow up, I guess. And these are the embryoid bodies that is how they would look like. So the next step is differentiation. Right, we want to get as from here to here. We have come till embryoid body formation, and now we need to go further. I don't think I will discuss too many examples, but we will see a few of them. There are also some biochemicals that we use within the culture medium. What we have to remember is, I don't have the pen, but maybe in the next class I'll try to describe it again. The cell differentiation, if we read it in cell biology or somewhere, a cell is influenced by two factors. One is the external factor, and one is the internal factor. Internal is the gene expressions of its own. External is what kind of contacts it has with its surrounding cells. What type of cells is it interacting with? And the other one is what kind of biochemicals are coming from outside, signals to the cell. So all these things will determine what the cell will become. So here, mostly because we are doing in vivo, we can play around with the different types of biochemicals or signals, biosignals we provide to the cell through the culture medium. Okay. So we have seen this. This is another picture of showing hanging drop method. It's just a close-up. I will discuss this in a little bit in a later chapter, a little bit more. But one thing is leukemia inhibiting factor. There is a biochemical that is present within the nutrient media. If you remove that, one should remove that to allow the formation of embryoid bodies, other than just giving the hanging drop type of method, or encouraging them to form interactions by itself and trying to make it as a sphere. The next one is conditioned media. It means you are, based on what type of cells you want, you have to make adjustments with the biochemicals in it, and only then one will be able to see derive different types of cells. You should also remember that differentiation of something like neurons happens spontaneously. That means you just don't need to do anything. You keep on culturing and remove LIF. They sometimes grow into neuronal progenitors and so on. Some cell lines come spontaneously, some will require conditioned media. We'll discuss more examples at a later time. Okay, here are some of the examples in which they say you have to be very careful about when we say about embryonic stem cells. Potential, it says. That means possible in the future, not yet approved for this thing. You should also remember that as I said, the approval is not given for most things that we know of. Majority of them are at clinical level. Keep that in mind. So if we have embryonic stem cells, we can derive any of these cell types: cardiomyocytes, neuronal cells, pancreatic islets, and so on. And these can be used to treat several of these diseases, like myocardial infarctions, the disease for the heart. You can use cardiomyocytes. Parkinson's, Alzheimer's, probably they can be treated with neuronal stem cells. So many others, these things. These are potential, means there are so many people having diseases of such as these, and we need to find a way to treat them. And through stem cells, these are possibilities, at least at the level of clinic. It is promising. Only thing we need to do more. And successfully being used at this point is for tissue engineering. If we are planning to make organs outside and then put it into the body, we need the tissue engineering comes in play with that. I think you are already learning, or you will be learning tissue engineering more when you go into your next semesters and so on. In that, you build a scaffold with biopolymers and add these cells. And the cells will start differentiating into that particular organ, or that is the goal, to be able to make embryonic stem cells differentiate into the cells of our desire, organize themselves into a tissue or an organ, and then transplant that organ into the human body. The best example or best use that it is already done is for pharmaceutical industries for screening of potential drugs. Say, for example, you take a compound. I think when you go into future with the research, you will read that they have taken a compound X, they added into and tested it on cell line Y, and it's not toxic, so it can be used in humans. But that's not true. It cannot be. It is not very toxic to that particular cell type. It doesn't define the rest of the cells, right? It doesn't say that one cell line doesn't represent all the 210 cell types in our body. But if you want to test in all cell types, then embryonic stem cells are a good source to do that. because you can derive any type of cell you want and then test the drug on it and see if it is going to harm the heart or ear or skin, anything. And that way, human embryonic stem cells are very useful, and many pharmaceutical companies already use it as well for screening of drugs, potential drugs, for toxicity and so on. There are scientific hurdles in the sense I want to use human embryonic stem cells, but there are also problems. The first problem is immunorejection. That means if we have one individual and trying to use some cells that are obtained from somebody else, then the problem is there will be immunorejection, which we discussed sometime earlier. So that is one first problem. Second problem is, say for example, I can't draw, but say for example, I had 10 embryonic stem cells, and I tried to make them or differentiate them into neurons, out of which 9 of the cells have differentiated into neurons, and one of it has not differentiated into neuron. These we refer to here as rogue undifferentiated cells. The other rest are differentiated. Now, if I put all the 9 or 10 cells into the body, the 9 neuronal cells may contribute to the repair of the brain, whatever disease it is. But this one cell may cause a lot of problems. That is, it can grow into an embryoid-like body within the human body, if it is transplanted into the human body, and that is called as teratoma. It's a tumor of cells containing tissues from all the primordial germ layers. You can see teeth here, you can see hair, and in some cases you might see rudimentary formations of eyes and so on as well. These are teratomas that might grow. Okay, so that is one serious risk people have or apprehensions about using human embryonic stem cells. And the third one is supply of cells. We need a large number of cells to treat any kind of disease, especially if it comes to glands and so on. And we do not have that much adequate supply of these cells. And the other one, if there is any, these are only scientific hurdles. There are other hurdles, like this technology would be expensive and it is time consuming, effortful, and so on. Okay, I hope you understood this concept, these two, very well. If you have any questions, we will repeat in the next class. So we will try to look at what are the remedies for immunorejection and what are the remedies for how to get rid of rogue undifferentiated pluripotent undifferentiated cells. The first one for immunorejection. Why are we talking about immunorejection? How can we get there is no way of getting the embryonic stem cells of a person that's already grown, right? We had this discussion also. If there is a human and the human is already grown, he must have grown from the embryo or zygote or the ICM. So there is no way of getting embryonic stem cells of this person. So we can only get embryonic stem cells from the discarded or disposed or donated eggs for research. And from there we get ICM derived embryonic stem cells, and we want to give it to this person, to this patient. So they may have differences in their immunocompatibility. And so immunorejection would happen. But what if there is a way of generating an embryo from this person and then derive embryonic stem cells and then give it to this person? That will be a good source of or good solution for immunorejection. So towards this, what we can do is somatic cell nuclear transfer. We will read about this in detail again later, but I hope you know or you heard the term animal cloning, which is somatic cell nuclear transfer. Everything is in the name. Somatic SCNT or somatic cell nuclear transfer. That means you take a nucleus from a somatic cell and put it into an egg cell, a nucleated egg cell. Let's see how it works. This is where we start. There is a patient here. Sorry, there is a patient here, and we take a cell from him, somatic cell, and then we take the nucleus and put it into the egg cell. The egg cell, the recipient egg cell, should be enucleated and should be of human. Some people have proposed that we could probably use that of animal. We will discuss that in a later time or in the later slides. So, enucleated means we have removed the nucleus or all the genetic material from the egg. And now we put from a donor cell which would have been in diploid, and this is 2n diploid, and you give electric shock, and there are other conditions that can increase the using strontium chloride and other things to activate this zygote. Excuse me. Sorry. Sorry for the disturbance. Yes. Are you all there with me?
Yes, sir.
Okay. Where was I? Yes, we can activate this zygote to divide further. I mean, you call it the zygote genome activation, and then the cell will start behaving as if it is a zygote and form blastocyst. And from the blastocyst, you can probably take human embryonic stem cells and then grow them on feeder layer like we already discussed, then differentiate using several condition methods and get whatever cell type we want and give it to the patient. There is no problem of immuno rejection. We are trying to see the SCNT method as a solution or remedy for immuno rejection. The goal is to generate embryonic stem cells that are patient derived so that they will be patient compatible, immunocompatible. I hope you know this as well. Animal cloning, Dolly the sheep, and many other animals have been cloned. And if you let this blastocyst grow and put it into a surrogate mother, it might divide, it might give rise to a baby, and that is nothing but cloning. Right? In animals, that is predominantly done. In humans, it is not allowed. Many countries have banned doing it. There will be socio-legal issues with doing that, and there will be a lot of problems because of that. But one can, with the consent, try to develop human embryonic stem cells that are derived from the patient, and then probably you transplant the derived undifferentiated cells to the patient. But of course, again, this is expensive and it will take a lot of problem. And there are many other problems too. Some of the problems with somatic cell nuclear transfer. We are trying to find a solution to a problem. Actual problem is patient has some kind of disease. So we want to treat the patient. So we thought about embryonic stem cells. But then embryonic stem cells have a problem with immuno rejection. So we are thinking about somatic cell nuclear transfer or animal cloning to derive patient-specific embryonic stem cells. Then again, we have problems with SCNT. And there will be many of those problems that will keep on happening. Say, usually this is how the suction pipette is. This is a suction pipette. It is an embryo. I'm just showing how an enucleated egg can be, or a somatic cell nucleus can be put into an enucleated egg. So the suction pipette will hold the embryo in place, or here it is the egg, for example. Then you take one of the somatic cell. I'm using a different example to show the technique part. This is a suction pipette, and this is the needle through which transplantation or insertion of the egg can be done, and so on. Okay. Yes. So we are looking at, I'll stop with this slide, try to understand how some of the problems with nuclear transfer or SCNT, somatic cell nuclear transfer, everything is in the name. First problem is efficiency. That means you take 10 eggs and you try to do this somatic cell nuclear transfer 100 times. Less than about 6% of them actually survive. Because as I showed here, we are rupturing the egg and so on, right? While we put, there are other methods where we don't have to rupture it as much. But still, putting the, during the nuclear transfer or somewhere, there could be damage, and because of which they do not survive. Not many would survive. So the efficiency is very less. The second one is epigenetic reprogramming. I will discuss this separately, and because we need to start from the fundamental level. Okay, and the other one is eggs needed. Who will give those many eggs? We say that about 6% are actually successful. In that case, where would we get 100 eggs, for example? That's very difficult. Nobody is going to donate. So humans, it's difficult to get human embryos, human eggs for these experiments. So some people have suggested animal eggs. So animal eggs can be used, but they are unethical. In the sense, some people may have problems with religious beliefs, or you cannot use eggs from pigs, or you cannot use eggs from cows. People will have different types of problems with that. Some other people like PETA may say you are actually manipulating the animals unnecessarily and you shouldn't do that. So there will be problems. There is also a biological problem or a technical problem, is that, say, if we had the egg in us, we have the nucleus, that is where the genome is present, and we also have mitochondria. Mitochondria has DNA. Mitochondria comes from the mother, whoever is the donor of the egg. Now, if you are going to put a somatic cell that is human, okay, diploid and human, and if this egg is derived from an animal, there will be discord between them. There could be. It's not that they have to be, in the sense that if you know that majority of the proteins that are present in the mitochondria are actually coded by the genome that is present in the nucleus. So the animal genome or animal mitochondria could be different from human mitochondria. Their requirements from the cell, from the nucleus. So that is what is differed here, disharmony between nuclear and mitochondrial genes. And if there is no insufficient supply or unnecessary supply of proteins that might be targeted to the mitochondria, then the mitochondria may not work properly. And if it doesn't work, then everything is the cell would die. The third, next, another problem is disarrayed mitotic spindles after nuclear transfer. As I discussed before, the egg is arrested in about start end of meiosis 1 or start of meiosis 2, and they are spent, the mitotic spindle is there, and you want to remove the DNA from here, then it is going to be difficult, and you're also spoiling the mitotic spindle arrangement that's already there. That may also cause, might result in the inefficiency part that we discussed here. The other part is similar, that are the misaligned chromosomes and aneuploidy. Aneuploidy is loss of some of the chromosomes. Say you're transferring in chromosome from outside into the egg, and during the transfer also some chromosomes could be broken, some could be dead as well. I mean, in the sense, some whole chromosomes could be lost, and that is a problem as well. I have a question here, and if you can answer this, we will stop here and discuss something else. Which is the best cell stage to be used in SCNT? What are the cell stages that we know of? We know G1, S, G2, and M. And we also know about something like G0. Okay, sorry about the handwriting here. So, which stage do you think is the best to be used in for SCNT? Anybody?
Is it M, sir?
Why is that?
It will give out commands for division rather than growth. So since we are taking from a somatic cell, it could be actually grown already, and we only need the division part now, so it can be M.
Okay. How would we transfer it? Say, during mitosis or meiosis, the nucleus is no longer existing, isn't it? The nucleus
Okay,
is,
yeah, it's
then
not intact.
that's right. Yes,
So it should be G1 because in G2 we already have two copies of everything, and
that's
it
absolutely correct. I mean, the thinking is right. G2 is 4n. Is that what you meant?
yes, sir.
Yeah, 4n. It will be tetraploid. That will be a difficult one. S phase synthesis is happening, so it will cause a lot of misalignment, and its cell will die basically if you transplant it. G1 is a good one, but the best would be G0. That is because in cell cycles, often what may happen, especially when you're growing in culture or so, is the G0, G1. We are not sure if it is finishing mitosis complete. It's everything is finished and perfectly stable, or if it is already entered S phase and so on. We may have those kinds of doubts. If there are some cells in G0, there we are sure that they are intact. Right? I think this is just a question to review several things that we have discussed so far. Okay, just give me a minute. I will try to check out some things for you. Yes. You can see my screen, right?
Yes, sir.
Yes. So we're trying to watch a video about how um, somatic cell nuclear transfer happens. It also can give you an idea about uh, how the ICM can be removed by mechanical dissection. It will give you an idea. There are several videos on YouTube you can actually check. This is an egg. It has the zona pellucida on the outer side, and this is the polar body. I think so. This is the suction pipette, which will hold. Can you hear any commentary? Can you hear commentary of the video?
No, sir.
No,
No,
sir.
right? Yeah. Find the link, or you can also find it how to do that. So you keep watching it. So that's a drilling pipette. There is a hole that is made somewhere here. It is removing and throwing here from the zona pellucida part. These are some parts of the chromosomes. This is the enucleated egg. Sorry, this is the enucleated egg. These are the parts removed. This is the nucleus from a different cell. Hole and new nucleus is being put into. It's amazing, isn't it? There are many techniques that you can actually check out and try to find. Let me see if there is any. These are general videos. I like to watch the video that I showed. I want to show you something, the video about. Used to have them. I changed my computer, so I couldn't get those. Let's see if we can find any videos on that. Okay. This sounds fine. Let's see. This is a blastocyst. You see the ICM here. These are the trophectodermal cells. Now you see this here. They're hitting it with a laser, so that the laser will make the hole in there. And then they are trying to push the blastocyst. There is a fluid filled. So I think what they're trying to also is disconnect the blastocyst from the zona pellucida. In mechanical dissection 1, we do not remove zona pellucida, if you remember. And now there is a hit here made, and a zona pellucida hole is being made. There is a hole made in the zona pellucida. You can think of some people can. You have to see it in science ways. You find it amazing to see that this technology is being. We can do so many of these things. Now see it as an individual, just as a human with a lot of feelings and so on. People can think, if we leave this embryo as such, it would have given rise to an individual, an organism. But now here we find it like a toucher kind of thing, isn't it? These are two ways of how you want to see things. Both are important, so that we will be able to do stuff that are important, but we'll also be will be responsible in the way we are doing stuff. Yes, it's like how many of you are. I think I will have 2 minutes after this video, and then I will ask. I think it's over time, so I hope you have seen that, right? You can watch many other videos that are present on YouTube. They will give you a very good idea about what is happening. Okay?
Sure.
Yes, any questions?
So is there a difference between SCNT and iPSCs, induced pluripotent stem cells?
Yes, they are different. As induced pluripotent, you take a cell and you make them pluripotent by genetic reprogramming, which we will discuss again later too. Yeah.
Okay, sir.
Here we are we are making a blastocyst and isolating ICM, and they are embryonic stem cells. Property-wise, we want them to be pluripotent. Okay. Yes, we'll discuss in the next class. Take care and have fun.
Sure, sir. Thank you.
Sure, sir. Thank you, sir.
Yeah. Bye-bye.
Thank you, sir.
See you.
Thank you, sir.